Hey guys, and welcome to your AMI day. If you saw my instructions about joining a Google Meet earlier this morning or late last night, please ignore them. There is no Google Meet today. Instead, there's two things on our assignment that I would like you to complete in order to be counted present. So they are two formatives. One of them is a concept check that we ran out of time to do last Friday. So on this concept check, you're going to click on one of these formatives and it's going to open up and it's going to say G9, so goal 9, CC for concept check, DNA to proteins. There are 5 multiple choice questions, almost said 6. The 6th one is just I answer the questions to the best of my ability. This basically helps us regrade this on the right scale. So I would like you to go ahead and answer that one. And then the other thing is a SNORC gene sequencing. I know, it's a fun name. So in this simulation, you're going to examine the DNA sequence of snorks. Snorks are an alien species discovered on the planet DNA in a distant solar system. You like what I did there? Huh? Snorks have only one chromosome with 10 genes on it. So just to let you guys know, you have a lot more genes, like a ton more genes. And your chromosomes are vastly more complex. And instead of just having one with 10 genes, you have 23 pairs of them with millions of genes. But because the snorks are simple creatures, we're going to pretend like this is how it is. So you've got 2 jobs here. One is something that we've already practiced. It is translating an RNA sequence into an amino acid. So we'll do the first one together. I've got a GUC. So I embedded these charts. And so if I start at G and then I follow it out to U and then I follow it out to C, and I see that I have the amino acid val or valine. So I'm going to go ahead and do this drop-down box and convert that to val. Now, something that we haven't done yet is talked about what those amino acids help do. So those amino acids are all going to form a long chain called a polypeptide chain. Polypeptide chain has different parts that are attracted to different sequences, and therefore they have a tendency to kind of ball up into a shape. That shape is going to be our protein, and that protein shape kind of works like a key to where it can start and stop certain things depending on which parts of the protein are exposed. This helps do a couple different things, but the main thing is that your proteins determine something we're going to talk a lot about in the next few weeks called your genotypes and your phenotypes. Your phenotype is what you look like. It's the fact that I have hazel eyes and brown hair and light colored skin and lots of other things. Not just physical traits, right? But also everything inside of you too, like the shape of your colon and how tall you are. So in our gene sequencing, we're going to look at some amino acid sequences, which will determine one of our SNORC's proteins. And these proteins, depending on what the sequence are, will determine their phenotypes and different things about them. This first gene that you guys are going to do will either determine if our SNORCs are hairy or hairless. And so as you can see, our SNORCs have a lot of diversity. Normally species do not have quite this much, especially not on 1 or 2 genes, but we'll talk more about that in a couple weeks. So depending on what this order is, whether it's Val, Ser, Lou, or Val, Ser, Liz, depends on if your SNORC has a hairy body covering or a hairless body covering. So we're going to do this for this SNORC. It should take you about 30 minutes or so. And then at the very end, you should have a pretty good picture of what your SNORC looks like. And I would like you to draw them based upon the traits that our Snicker SNORC had. This is the Snicker SNORC. There's 3 other SNORCs, but I thought maybe that was too many SNORCs. And now I'm just kind of stuck saying the word SNORCs over and over and over again. If drawing on formative is not your jam, then you can get a piece of paper at home. If you have some paper at home, hopefully you have some paper at home, and draw a picture of what they look like. Otherwise, I would just recommend just freehanding that sucker in Google Draw. Enjoy. You guys can always email me if you have any questions, or I can hop on a Google Meet with you and answer some things.